Hello students, welcome to Biotech Academy. Today we'll continue with our discussion on protein folding and misfolding. In my previous lecture I discussed about the classical and Anfinson's experiment, who was the first to demonstrate that correct folding is absolutely essential for the protein to perform its specific task. So today we'll discuss how the process occurs, how the entire process occurs, what are the processes, what are the methods are involved in protein folding, and how misfolding may result into various disease manifestations. Alright, so the process of protein folding provides an elegant example of biological self-assembly. Means that how it provides an example of how the cell, within the cell, the entire process of protein folding, starting from its protein synthesis to protein folding to perform the specific task, which the entire process, how the cell governs the entire process from synthesis to protein folding, how the process works, so that, and how accurately the cell performs, so that the chances of error are very less over by the cell. The chances of making a cell any kind of an error is very less. So the accuracy is very high by the cell for any of its function. And understanding this complex machinery, there are various steps are involved, and it is a very complex procedure from synthesis to modification and to correct folding into its tertiary or quaternary structure. So this is the entire process is highly complex, and understanding this complex machinery, it has been a major research area from several decades in both biology as well as chemistry, because various types of bonds are involved, various types of structures are involved. So chemistry is also, lot of chemistry is involved over here, biology is involved. So the entire process, understanding the entire process, is a very major research area, and it has fascinated many scientists across the globe, and they are working hard to deduce the entire process with how the processes are occurring in the cell for various types of proteins, because the cell makes thousands of proteins, and each protein has a specific tertiary or quaternary structure which it attains for correct functioning. So whenever a new protein is identified, so what is the structure, how it may have attained, what is the sequence of this protein, how folding has made a compact structure. So the entire processes are being discovered by many scientists and researchers across the globe. So in protein folding, the first important thing is that the amino acid sequence, it plays a pivotal role in determining what kind of secondary, tertiary or quaternary structure the protein will attain. So apart from amino acid sequence, now when we discussed, if you remember, various kinds of interactions which are involved in determining the different levels of protein structure, there are certain specific types of bonds which are helpful in attainment and stabilization of the three-dimensional structure. So the process of protein folding is governed by the distribution of polar and non-polar kind of amino acid residues. Why the distribution is essential? Because these different kinds of amino acid residues, which are different because of their variable R group, the kind of interaction that these amino acid residues will form is dependent on the R group. So their distribution determines what kind of bond it will form, and because of those bonds, what kind of tertiary or quaternary structure that particular protein will form. Now, if you remember, when we talked about the various amino acids, we did speak about the arrangement of polar and non-polar side chains. So the polar or the hydrophilic ones, the amino acids with the R groups which are polar or hydrophilic, means they are water loving, they tend to gather on the outer surface of the protein structure. Why? Because the cell is filled with water, and if the non-polar residues will be on the outer surface, they will not, because they will repel the water moieties and the structure will not be stable. So in order to stabilize the structure, the protein folds and arranges itself such that the polar residues, they are on the outer surface, while the non-polar residues, they are arranged inner, so that these non-polar residues, they are in minimal contact with water, whereas the polar ones are on the outer surface and they form hydrogen bonds with water and stabilize the tertiary structure. So now, you must be aware of the names of polar and non-polar amino acid residues. When we discussed about amino acid residues, I also described about the classification of the amino acids. When we discussed about the properties of the amino acids and mentioned some of the important names of the amino acids, you must know some examples of polar and non-polar residues. So also, if you know the protein sequence, because the amino acid sequence determines the bonds which it will form and dictates the protein folding process. So if you know the amino acid sequence of a particular protein, then you can postulate or you can hypothesize what kind of secondary, tertiary or quaternary structure that protein will form, and because of the properties, basically because of the properties of the R groups. Okay, so it is entirely dependent on the amino acid sequence. Now, but important thing you must know over here is that the hydrophobic residues can drive a what you call as hydrophobic collapse, because what is hydrophobic collapse is that when these hydrophobic amino acid residues, they come very close to each other, then there is loss of water which are surrounded by these amino acid residues, and the entropy of that particular system is increased. So that is called hydrophobic collapse. So this is one of the important thing you must know about hydrophobic collapse. In this so hydrophobic amino acid residues name you must know, because the questions are asked what there will be in the options. You can have various amino acid residues. You'll have 4 amino acid residues, out of which only one will be hydrophobic, and the question will be like which of the following residues can cause hydrophobic collapse. So only if you know the hydrophobic amino acid residues name, you'll be able to answer this question, and this question is very particularly important from GATE point of view. So those who are targeting GATE, so this point is very important for them. Now, in the previous lecture, we did talk about Anfinson's experiment. Alright, so that experiment was the first experiment which provided evidence of that correct folding is absolutely essential for the protein to perform its specific task. And in that experiment, Anfinson used certain denaturing conditions, certain agents which denature the three-dimensional structure of the protein, like urea, beta-mercaptoethanol, or guanidinium hydrochloride. So in the presence of these denaturants, the tertiary structure of the particular protein which he was targeting, that was ribonuclease, so that the three-dimensional structure was lost, and upon removal of the denaturing conditions from the protein again refolded into its correct form. So when you are isolating protein from a particular cell and you're providing denaturing conditions, you will get a denatured protein or an unfolded protein. However, upon removal of the denaturing conditions, the protein again has the capacity to refold into its correct form and perform again. And again, if you study the function of the protein in vitro, you'll be able to see that particular protein has folded into its correct form and is performing its task accurately. So basically, if you can see, this is the tertiary structure, this is the 3D structure of a particular protein, where there are disulfide bridges, there are hydrogen bonds, there are hydrogen bonds, disulfide bridges, then there are hydrophobic interactions, and there are many other interactions, and there are van der Waals forces. So these are main interactions which are stabilizing the three-dimensional structure. And if you are providing certain denaturing conditions like high concentration of urea or guanidinium hydrochloride, or in particular if you are increasing the temperature, so if you are heating the protein mixture, then the protein will lose its structure and it will form a linear polypeptide. However, if you are again cooling or removing these denaturing conditions using dialysis process, then the protein again has the capacity to reform into its correct three-dimensional structure. But this process takes time, and you have to allow the protein, you have to give the protein in vitro certain time so that it falls into the correct form. So basically, there exists two forms. Either the protein will be in its three-dimensional conformation, or it will be in the correct folded form, or it will be in unfolded form. So basically, the graph, if you plot a graph, it will look like these are the two axes. On x-axis we have denaturants, and on y-axis we have unfolded proteins. So in the presence of denaturants, gradually the protein denatures or unfolds, but the initial process, it is slow. However, when the denaturing conditions or the denaturants, the concentration of these increases beyond a certain limit, then the rate of unfolding or the rate of denaturation increases to a very great extent, and the rate of unfolding increases at an exponential level, and then again a threshold level is attained. So basically it is an S shape, where initial at lower denaturing conditions the unfolded proteins are very low. However, if we are increasing the denaturants to a greater extent, the rate of unfolding or the rate of denaturation increases such that the most of the protein unfolds or denatures. And then even if you are increasing the denaturing conditions or the denaturants, if you are increasing the concentration of say urea or guanidinium hydrochloride, still most of the bonds which are stabilizing the structures, most of the bonds which are stabilizing this structure, this folded protein, are broken, and so a threshold level is attained. So this is basically how the unfolding occurs. So there are two forms. Either the protein will be present in its correct folded form, or it will be in its defolded or denatured protein. Now the question is, how does this process of protein folding work? So various hypotheses and various mechanisms have been proposed to explain the process of protein folding. As the process of unfolding is cooperative, what we have discussed over here, that there occurs this transition from folded to unfolded when we increase the denaturing conditions or the denaturants. So there occurs a sharp transition from folded form to unfolded form. Similarly, so this is a cooperative process. Similarly, the process of folding is also cooperative. Now, you remember we have discussed about there are various types of interactions which are driving the process of folding and stabilizing the folded structure or the native conformation of the particular protein. So if you look at each interaction individually, they are weak. However, it is the cumulative effect, but because there are numerous kinds of interactions and there are numerous types of greater number of bonds are present. So it is the cumulative effect of the total number of weak interactions which impart stability to the folded form, and the completely folded form is also called as the native conformation of the protein. Once the process of folding starts, once the protein, after the synthesis of the linear polypeptide chain, the process of folding starts, there are numerous intermediates which are formed between the folded and the between the completely unfolded or the linear polypeptide to the completely folded or the native three-dimensional conformation of the protein. There are a number of intermediates and there is a decline in the entropy of the system. So initially when the protein is synthesized by the process of translation, so that is a linear string of amino acids, and there is maximum entropy or there is maximum free energy of the system within the system. So but that amount of that free energy, larger free energy or greater entropy, it is not stable conformation. So once the protein starts folding, there is a gradual decline in the entropy of the randomness in the system, and when between the intermediates, there are when the folding occurs and the compaction of the polypeptide is going on, so there is a decline in the entropy, and the final native three-dimensional conformation, completely folded form, it has the minimum entropy of the system, so that form is the stable form. Now the next question, that is, how do we predict the structure? So how do we predict that particular, say a particular protein A which has approximately it has 30 or 40 amino acid residues or even more, and you know the sequence of the residues, all the 40 amino acid residues, starting from the N terminal to the C terminals from the first to the last, the correct order in which these residues are present. So now you need to predict what kind of secondary, tertiary or quaternary structure that particular protein will attain. So how do we do that? So if you remember, when we discussed about the properties of amino acids, and when we started with our discussion on various levels of protein organization, we discussed that there are certain residues which favor a particular kind of conformation, a particular kind of secondary or tertiary conformation, and however there are certain residues which try to create a kink between that structure, three-dimensional conformation or the tertiary structure. So it is totally dependent. There is a very intricate sequence and structure relationship. So if you know the sequence of the amino acids, you can theoretically predict the secondary or tertiary structure that particular protein will attain. So say if there are proline residues, they will form beta sheets. If there are glycines, they will favor alpha helix formation. So likewise you can predict, that is theoretical prediction of the proteins. However, during the actual process of protein folding, there are various other players which determine which modify the interactions. So that is theoretically you can predict, but there are other factors also which might turn the entire conformation into some or the other way which you have predicted. But ab initio you can definitely predict from the sequence of the amino acid residues. If you know the sequence of the residues, you can predict what kind of secondary or tertiary structure that particular protein will attain. Now we'll come to the thermodynamics of protein folding. So folding of protein into the native conformation, it is a spontaneous process, and it occurs under physiological conditions, and it is basically again and again I am emphasizing that the folding process is dictated by the primary sequence or the sequence of the amino acid residues or the linear chain of the polypeptide. So the process of folding is thermodynamically favorable process where gradual decline in the entropy of the system occurs from an unfolded linear polypeptide to completely folded protein, three-dimensional structure of the protein. So the linear polypeptide, so say suppose this is a linear polypeptide and all these circles, they represent amino acid residues. It has very high, or you can say it has maximum entropy or free energy. It has very high free energy. Then it gradually starts folding into specific conformation. So there are various bonds which are formed. So there is a decline in entropy. So the intermediates, all the intermediates, as these intermediates move towards the final native conformation. So a native conformation like this has arrived. This is the native conformation of the protein. So all the intermediates which are towards the linear polypeptide chain will have lower entropy as compared to the linear chain, and the polypeptides and this native conformation has the minimal entropy or minimal free energy, and it is thermodynamically favorable. And because of the minimal entropy, this conformation, this native three-dimensional structure is highly stable. So this is thermodynamically favorable process where there is gradual decline in the entropy of the system once the process of folding occurs or once the process of folding starts. The thermodynamics of protein folding by this open funnel model. So this open funnel model, so how does the funnel look like? Looks like it has a wide open mouth and a very narrow bottom end. So initially when the protein is synthesized, it is a linear string or a linear chain of amino acid residues, which is the primary structure of the protein. So over here the entropy is very high, and it is thermodynamically very unstable molecule because of the high entropy or high free energy of the system. And these arrows represent the high entropy, and over here these linear polypeptides or the primary structure, they lie at the mouth of the funnel or the open wide end of the funnel. Now as the amino acid residues, the side chains of the amino acid residues, they start interacting and they start forming hydrogen bonds, and to initiate the folding process there occurs a decline in the free energy, and between the inner region, this slightly narrower region, it represents the intermediates of the folded protein where the entropy is comparatively less as when we compare the entropy of the linear polypeptide chain or the primary sequence, and it is the end or the narrow terminal of the funnel where the completely folded three-dimensional conformation or the native conformation of the protein exists, and this has least entropy, and it is thermodynamically least entropy, thermodynamically highly stable. High entropy, highest entropy, maximum free energy, and thermodynamically very unstable molecule. So this is how the protein fold, the thermodynamic, this is where how we can describe the thermodynamics of the protein folding. Now we come to molecular chaperones which are involved in protein folding. So we are now talking about chaperones. And what are these chaperones? These are certain protein systems which enable or which guide the process of protein folding, and it allows the protein, the linear chain or the primary structure to attain its correct folded form. So these are, and they also prevent aggregation and misfolding of the proteins within the cell, because they can lead to the aggregation of misfolded proteins, may lead to certain diseases which we'll discuss at the end of the lecture. So these are the chaperones. There are specific chaperones, and a wide variety of the first chaperones was studied in E. coli, and there are various chaperones which have been identified in E. coli, and they are GRO-EL, DNA-J, DNA-K, GRO-ES. So these are certain chaperones which have been identified in E. coli, which is the model organism. And the bacterial chaperonin, which is GRO-EL, this GRO-EL, it binds to the proteins in its unfolded form. So it binds to the linear, basically it binds to the linear polypeptide and starts, and then it guides the polypeptide to attain its correct folded form. And this, once the protein has attained its correct conformation, or it has attained its native conformation, this enzyme, this GRO-EL complex, it can be attained back into its the entire quantity which was bound to the unfolded protein. The entire quantity can be recovered back at the end of the process when the unfolded protein has completely folded into its correct three-dimensional conformation. Now I'll discuss how the process of our folding is guided by these chaperones. So initially there is a linear polypeptide. This is a linear polypeptide, or you can say it is the primary structure or a primary sequence, or basically the unfolded protein. And these chaperone proteins, they start to interact with this unfolded protein. So basically first DNA-J and DNA-K, these are ATP bound proteins which gets attached to this unfolded protein. So what we have DNA-J and we have DNA-K over here, which are attached to this linear polypeptide chain, and there is ATP which is bound to these proteins. Okay, now hydrolysis of ATP occurs, and this hydrolysis of ATP leads to formation of ATP and inorganic phosphate, and this hydrolysis is caused by DNA-K, and this hydrolysis is caused by this hydrolysis is caused by DNA K, but it is stimulated by DNA. This is stimulated by DNA J. Okay, so this hydrolysis is caused by DNA K. Basically, it is carried out by DNA K, but what guides DNA K to carry out the hydrolysis? It is DNA J, which is dictating DNA K to carry out the hydrolysis of ATP. Now, this DNA K and ADP complex, it is still associated with the unfolded proteins. This is tightly bound to the unfolded protein. So what we have DNA K and ADP now, because ATP has been broken into ADP. So this is still tightly bound to the unfolded protein. Now there comes another factor, which is GRPE. This is a nucleotide exchange factor, GRPE, which comes over here, and it facilitates the release of this GRPE, facilitates the release of ADP with DNA J, and DNA J is also over here. And what it does, it causes the release of these two molecules. So we have the polypeptide with DNA K only. Okay, so now we have this polypeptide, which is partially folded with DNA associated with DNA K. So this DNA K remains bound to the partially unfolded protein, and the process of protein folding continues to attain thermodynamically stable conformation, which has less entropy. And then once the protein gets completely folded into its final three-dimensional conformation with minimal entropy, then only DNA K gets detached, and it is then again bound to the free ATP within the system, and it is charged again to carry out another process of another round of protein folding. So it is recycled once by getting attached to ATP which is freely available within the cytoplasm of the cell, once this polypeptide has attained its final three-dimensional conformation. And during this entire process, if there are any other protein molecule or which may not have attained its final conformation, then that unfolded protein is taken up by GROEL. So that unfolded protein molecule is taken up by GROEL at the end of this cycle. That is taken up by GROEL, and then this system, this GROEL chaperone system, it completes the cycle of that particular unfolded protein into the folded and makes that protein attain its final folded conformation. Coming to the last topic for this lecture, that is protein misfolding. So we'll discuss about protein misfolding over here now. So we've discussed that accurate folding is absolutely essential, or attainment of the correct three-dimensional conformation is absolutely essential for the protein to perform its designated task. And if that particular protein, it does not attain its native conformation, or it leads to misfolding, some error because of certain errors, when it does not attain its final conformation, then the function associated with that protein is lost. So we lose the function of the protein once it does not attain its final three-dimensional conformation. So that is because of the misfolding of the protein. So basically, these misfolded proteins, the aggregation of these misfolded proteins, they lead to various disease manifestations within humans. So for example, Alzheimer's, cystic fibrosis, pulmonary emphysema, etc. These are certain diseases which are caused because of the protein misfolding. And each protein is destined to attain a particular three-dimensional conformation, and there are certain mechanisms by which the misfolded proteins are removed by the cell, so that the waste material does not accumulate. However, there are certain conformations, certain protein conformations, misfolded conformations, which still accumulate within the cell, and they may lead to disease manifestation. So there are three types of fate of the newly synthesized protein. So if we have a newly synthesized protein, basically it is a linear polypeptide which is obtained at the end of the translation process, or you can also say that it is the primary structure of the protein. So the first thing is that it is correctly folded. It attains its correctly folded form without any help, or it is folded into correctly folded. The protein is correctly folded with the aid of the chaperones, which we discussed just now. Okay, then the third one is the incompletely folded form, or the incompletely folded form, which is degraded by the incorrectly folded forms, which are degraded by proteasomes. Apart from these three fate of the linear polypeptide, then the last one is aggregation of misfolded proteins, and it is this aggregation of the misfolded proteins which causes disease manifestations within the cell. So because the normal functioning of the protein is lost upon misfolding. Alright, so this is all about protein folding and misfolding, and we'll continue now in the next lecture. I hope you all have found the content useful, and if you all have any doubts, please ask in the comment section, or you can send me the mails. There are those who are sending mails. I'm continuously replying to them. So I encourage you to so that you can clarify your doubts, go through the books, go through the reference books, and still if you face certain difficulties, you can ask me. I'll feel privileged to answer all your doubts and clarify all your queries. So thank you.